regulatory sequences, epigenetic changes, cell differentiation, and sequential gene expression all lead to the phenotype of a cell or organism. So when we start talking about genetics, we aren't just randomly doing transcription and translation, and it's not happening all the time all at once. We begin with the chromatin structure, and then there's transcriptional control, and there's post-transcriptional control, and then there's translational control and post-translational control. And there's regulatory events that occur at all of these different stages. So we're going to start at the chromatin structure. So when we start talking about the epigenome, we're talking about not necessarily the actual genes, but what controls which genes are expressed. So your DNA is wrapped around histone proteins, and we call this complex a nucleosome. And the histone proteins have these tails that hang off. So when the DNA is wrapped tightly around the histone proteins, promoters and genes might be inaccessible because they're wrapped too tightly. So you can have these different complexes that can get involved and loosen up the coils in order for promoters and the genes to be accessible. So another way to look at this is, again, we start out with all the chromatin tightly wrapped around the histones, and then you can have acetylation. And acetylation is one of the groups that can be attached to the histone tails in order to loosen up the coils. And there is an enzyme that helps make this occur called the HAT enzyme. So when you have a lot of HAT, you have a lot of acetylation, which makes genes more accessible. So again, these histone tails can have acetyl groups attached to them and make the genes active, or at least make the DNA accessible for the transcription factors. Now, this process is also reversible, so you can have deacetylation through the activity of a different enzyme, the HDAC enzyme, which then would remove the acetyl groups and tighten back up the chromatin structure. Another thing that closes off the chromatin structure and makes DNA inaccessible is methyl tags or methyl groups. So these methyl groups actually attach directly to the cytosine base and not just to the histone tails. So the methyl groups are going to attach to these cytosines, and that's again going to have the DNA wrapped tightly around the histones. Now, making genes inaccessible doesn't necessarily mean that's a bad thing. As cells differentiate and become different types of cells, they're not going to make proteins associated with other tissue types. They only are going to express the genes with that particular tissue type. So methylation does happen naturally in a person's life, and especially early in tissue development. But methyl tags can also be caused through toxin exposure, changes in diet, good or bad, exercise, but also like trauma and stress can also affect methylation. So again, if you had identical twins, they would have the exact same genome, but that doesn't necessarily mean that they have the same epigenome. So at 3 years old, almost all of the same epigenetic tags are in place. But as a person ages, and these same twins are looked at as 50-year-old, all the red and green spots show where their epigenomes are different. And again, if the epigenetic changes are affecting expression, you're going to have different expression if you have a different epigenome. If you have different expression, you're going to have different structure because you're going to be making different proteins. If you have different structure, you're going to have different function and ultimately different phenotypes. So again, going back to control and regulation, we just got done with the regulation at the chromatin structure. There's also, again, transcriptional control, post-transcriptional control, translational control, and post-translational control. So now we're going to focus on the transcriptional control. And transcriptional control gets down to regulatory sequences. So you have the actual code for the protein as the gene, but upstream we have promoters and enhancer regions that are also going to be considered regulatory DNA sequences that control expression. So again, in order for a protein or transcription to occur, we first have to have activators land on enhancer regions. So again, these activators can be hormone receptor complexes, which again is going to tie into proteins. We also have transcription factors that bind to the promoter regions, and RNA polymerase is also going to hook onto that promoter region. So again, the transcription factors are also proteins, and they're going to aid in that recruitment of the RNA polymerase, make it hook on a little bit better so that more transcription can occur. But again, these activators have to be brought over to the promoter region to kick off actual transcription. So again, that transcription is going to lead to the production of messenger RNA, and that's going to ultimately lead to the translation of proteins. So when we start thinking about this, again, we have gene expression. All of these things within eukaryotes are necessary for a gene to be expressed. You have to have the activators on the enhancers, and you have to have the transcription factors on the promoters. So we can have this sequential activation, especially in development, which is really important. So the presence of activators and transcription factors leads to target gene expression. So again, these transcription factors and activators are going to be associated with the actual transcription of specific genes, and that target gene expression can lead to more activators and transcription factors, and very specific ones. So again, remember, these transcription factors are proteins, and the receptors that bind to the hormones for the activators are also proteins, which means that to make more transcription factors and promoters and receptors, or for different ones, you have to turn on certain genes. So the presence of these new transcription factors will lead to more target gene expression, and then that could lead to more activators and transcription factors for other genes, target genes. And we have this sequential activation. That's really important in development because certain genes need to be turned on in a certain order. Now again, these target genes can lead to more or different transcription factors, but you're also going to lead to specific traits and phenotypes that come about from those targeted genes. So we have the same genes. If inside every single cell in your body, you're going to have the exact same genes, but you might have different expression. So in every single cell in your body, you're going to have the gene for albumin and the gene for crystalline. However, not every single cell in your body, and you have about 200 different types of cells. Those different cells are going to actually transcribe different genes. So we call that differential gene expression. You have the same genes, but different cells are going to express different genes. And that comes down to a lot of what are available as activators. So if we look at like a liver cell and a lens cell in your eye, within that nucleus, they're going to have different activators present. And with these activators, they're going to bind to these enhancer regions, and therefore in the liver cell, they're going to make albumin, which is a blood protein. However, in the lens in your eye, we have different available activators, and these are going to bind to the crystalline gene, which is going to add to the structure of the lens. So your liver does not make crystalline because it doesn't have the activators available to make that gene. And your lens is not going to make blood proteins, albumin, because it doesn't have the activators for this particular gene. So when we talk about this, we we have tissue-specific proteins because we have differential gene expression. Last example here, how gene expression can lead to different phenotypes. When we think about calico cats, they're always going to be female, and they're going to be heterozygous for black and orange color. Now, within female animal cells, they have two X chromosomes. However, only one X chromosome is active inside every single cell, and randomly we have deactivation or inactivation of one of the X chromosomes. And in development, wherever there is black fur on the cat, the orange X has been inactivated, and it's going to express the black fur color. However, wherever there's orange fur, it's going to have inactivated the black X, and it's only expressing the allele for the orange color in the orange patches. So again, if you express different genes, you're going to affect the phenotype. So again, we have regulatory sequences, epigenetic changes, cell differentiation, and sequential gene expression, all leading to phenotypes that are unique to cells and organisms.